Thank you so much, Tina, for inviting me. I want to thank all of you for coming to my talk. I'm really honored to be here. We consider UW to be, our premier site of laboratory medicine, and so I've been having a great day meeting people both in research and clinical areas, and it's been really a lot of fun. And so I'm excited to talk to you about the work that I do in my research lab. As Tina alluded to, I've been interested in gene deregulation in hematopoietic malignancy for many years now, and have focused particularly on histone modifications in terms of epigenetic regulation. And then more recently have started looking into long non-coding RNAs, and so I'll tell you about that a little bit. So I have nothing to disclose. So as I said, my lab is interested in the molecular mechanisms that drive gene regulation and deregulation in particularly lymphoid cancers. And we approach those questions with a variety of different methods, including very global methods of epigenetics and RNA-seq, and then also more targeted methods of trying to develop novel therapeutics that reverse abnormal epigenetic modifications at oncogenes or tumor suppressors. So just a little bit about why we study lymphoid cancers. I sort of cut my teeth, so to speak, on myeloid leukemias. But the reason that lymphoid cancers have been a great model for me for epigenetic regulation is, particularly as compared to leukemia or many other solid tumors, is that lymphoma patients are sort of unique from a scientific perspective in that oftentimes the malignant cells are confined to a lymph node, but yet peripherally they have normal cells of a similar lineage and stage of differentiation. And so when you're looking at epigenetic modifications and looking at expression profiling, to have a normal cell from a matched patient is incredibly powerful for understanding what's really going wrong. And so that's one of the major reasons driving my interest in lymphomas. It's also a fairly common cancer, affects increasingly more people as we all live to older ages. And of course, helpfully there are models in mice and in cell models that we can do experiments in. So I'm going to talk to you today about 3 different projects that are in the lab. And they span B and T cell lymphomas and different gene regulatory mechanisms. So the first that I'm going to talk about is super enhancers in B-cell lymphoma. I wanted to give you an overview so that you can kind of get an idea about how we do things in the lab, because it carries over into each one of the projects. And so we take a global approach to begin with, and we do discovery omics analyses, whether it's ChIP-seq, RNA-seq, exome whole genome, what have you, in primary patient samples that we collect with the help of our collaborators in oncology. And we look at genome architecture, so chromatin looping, as well as histone modifications, and then, of course, transcription. And so we have patients and we have normal controls, and we do all these omics analyses. And then we also create tools to help us to analyze this data. We're not a native informatics lab, but we realized that the available tools were not doing what we needed them to do. And so my students, who are molecular genetics and genomics graduate students, took it upon themselves to then code and develop the tools that we needed for our specific questions. And being students, they came up with some very creative names. Here are 3 of them: Plato, Genotify, and Vampire. And I wanted to put in a little bit of a plug actually for Genotify because of the audience that I have here. It actually, I think, would be helpful for those of you who may be looking up genes, whether they've come up in some panel that you've done or some paper that you're looking at, and you're like, gosh, what does that do? And you have all these different websites that you can go to. But then you have to go to all these different websites, right, to tell you about the gene. Whereas Genotify is like one nice little package. You can put it on the side of your screen, and it will tell you everything you need to know about the gene, including its expression in various tissues in humans. Anyway, it's very nice. So I can give you the GitHub link if you're interested. And it actually came out in the Journal of Open Source Software, which is, the acronym is JOSS, which I think is fun, this summer. So that was an interesting experience. We had never published in an open source kind of where my student actually interacted in a chat with the reviewer in real time. And they were known to each other, picture, name, everything, just going back and forth. Oh, I think you should do this. Oh, cool, okay, I did that. And oh, I think you should do this. Well, I couldn't do that for these reasons. Oh, okay, that's cool. The whole thing took 7 days. It was amazing. It was a good experience. I recommend it as a journal. Anyway, so we take all of these studies here and we try to distill it down to loci that we can actually then functionally characterize. And we're looking at enhancer function. We can use CRISPR, knockout, knockdown, and luciferase assays to query those, looking at lincRNA function with similar kinds of approaches. And we use cell lines and primary patient cells as our models for the most part, although we're starting to get into PDX lines. And then, of course, our wonderful in vitro kinds of studies, molecular biology and biochemistry. So just as, so that kind of we're all on the same page, mechanisms of gene regulation, the ones that I will start talking about are at the level of histone modification. So of course we all know that DNA is packaged around nucleosomes, which are made up of histones, and those histones have modifications. Depending on the modification, that is a signal then to how highly the gene should be expressed. So for example, H3K4ME3 is an activating mark that is especially at promoters. And K27ME3 is a repressive mark that tends to be in very repressed regions, including heterochromatin. One mark that is not here in this particular cartoon, but maybe over here I can't really see, is H3K27 acetylation, which is an activating mark also that is indicative of activating enhancers, which is one of the things that we're very interested in. So in general, for the purposes of my talk, not the purposes of epigenetics as a field, because it's too complex to go through all of it. In the purposes for my talk, if I talk about acetylation, it means activation, okay? And ME3 is also activation. If I talk about K4ME1, it's just a mark of enhancers that will just be present in any enhancer, whether it's active or not. So I just wanted to sort of give everyone that baseline before I show some of the data slides. So as I have sort of already alluded to, we have patients who are, we get biopsies, lymph node biopsies, or we do have a project in cutaneous T-cell lymphoma, which then we get skin biopsies, and sometimes also tumor or lymph node biopsies from those patients. And then from the controls, we get peripheral blood, but also tonsils. So they're a very good source of activated B cells. And then our various omics, which what I haven't mentioned so far is CTCF chip, which delineates the boundaries of active or repressive chromatin, and also ChIA-PET, which is a way of looking very specifically at interacting loops of DNA. And then Hi-C, which is a more global way of looking at interacting boundaries of DNA. And then, as I said, those are the tools that a few of the tools that we've created. So one of our initial discoveries was that we can look at these enhancer profiles, and we started with follicular lymphoma, which is a very indolent lymphoma and hasn't gotten quite as much research either clinically or in the basic science field. Partly because it takes the patients like 20 years usually to pass away, and so trying to actually establish any clinical correlates is somewhat challenging. But follicular lymphoma is incredibly common, and there are some patients who don't do well, who progress more quickly and don't respond to the therapies that, say 75, 80% of the rest of follicular lymphoma patients respond to. And so the surprising thing that we found in this initial study, and this was unbiased, and I know it was unbiased because I was the one who actually did this particular analysis, Because if my student or postdoc had come to me with this separation of follicular lymphoma patients, comparing them to normal germinal center B cells or activated germinal center B cells, and shown me this, particularly in follicular lymphoma, I would not have believed them that they had found it in an unbiased fashion. But in fact, I saw this pattern no matter how many different ways I analyzed the data. And as it turned out, the genes that were located near these enhancers, okay, these are all enhancers, and this is K27 acetylation, all of them were, or a great majority of them were activated, as you would expect from active enhancers, but showing that this enhancer pattern was actually functional and was leading to gene expression changes that we could detect. And then the other thing that was surprising was that these were also the patients, these few patients were the ones who also progressed very quickly and didn't respond to therapy and ultimately had poor outcomes. And so that was a really exciting finding for us, that you could see this pattern even in the enhancers. So from that, I want to move on to then a follow-up study that we're doing that isn't published yet. And so I don't know how many people in the room are familiar with the term super enhancers. Some people really hate the term because they think it's just a rebranding of an element that we've known for decades, which is locus control regions. And I'm not particularly married to any term, but these are clusters of enhancers that are very, very strong and tend to drive tissue-specific expression of very important genes and often drive the expression of oncogenes in certain cancers. For example, MYC has a massive super enhancer. So if you're more of a visual person, this is cartooned here, where the typical enhancers are not that different between these two cell types, but the super enhancers are very different. And then the genes that whose expression they're driving are much more highly expressed. The other thing that you should know about super enhancers is that they often are driving a large number of genes, and they're in very gene-rich regions, and so they can be hard to dissect individually. If you want to understand the function of a particular super enhancer, that can be challenging because there are many genes that they're driving. So in this particular study that we did, we had K27 acetylation ChIP-seq from actually many different types of lymphoma, so follicular lymphoma, DLBCL, and also chronic lymphocytic leukemia lymphoma. And we, my student Jared, did defined the super enhancers. So this is not the way that super enhancers were initially defined, but the way that they're typically defined now is by basically summing up the amount of K27 acetylation, that activating mark that is in enhancers, and then just ranking them. Like, what could be easier, right? You just rank them on this nice little plot. And if you draw a line at the inflection point of this, which in the lab we call a swoosh, very, very scientific, I know, then these are considered to be the super enhancers. It's somewhat arbitrary, but if you actually then go and look at the genes that are driving the enhancers that are in this grouping, they are lineage defining, they are oncogenes, and so it really does practically work. So Jared designed this awesome super enhancer calling pipeline before he found that there was some software came out later that he could use, but he designed all of this pipeline himself, and it worked really well. So we had some criteria, as you need to, since there's so many enhancers, and we can only functionally characterize a small number in the lab. So we really wanted it not to overlap a gene, because if we're going to use CRISPR, we can't be cutting out the gene. So obviously it needed to be recurrent across lymphoma. And it had to be different from normal B cells because, we study lymphoma. And then, obviously, it had to have an apparent functional impact on gene expression. And once we got through all those criteria, there were fewer than 50 super enhancers to look at. So one of these super enhancers that I'm going to talk to you about today it was ranked here in the swoosh, so not the highest, but fairly high. And it is between FCMR and PIGR, which are both Ig receptors Those who are immunologists in the room probably recognize, most people call it PIGR, and it's actually a really cool protein, I think, in that it is only expressed in epithelial cells, and its job, so in the gut, for example, is there are plasma cells, right, that are pumping out antibody, and this PIGR, this receptor, is expressed on the basolateral side of the cell. It grabs those antibodies and it transcytoses them across the cell and then releases them into the gut to clear bacteria out of the gut. I think that's really cool, but most of my immunologist friends don't think that that's as cool as I do, maybe because it was discovered too long ago. I don't know, but I think it's cool. So for the other interesting thing for me about this locus is that this is less than 5 kb in humans. Okay, the locus is very similar in mice. The space is a little bit bigger. But this protein, this is a IgA primarily receptor. This is a primarily IgM. This is expressed only in B cells. This is expressed only in epithelial cells. And yet, much of the locus looks the same, including many of the enhancer profiles look similar. And if you look in the literature, there are of course very divergent literatures, show the same transcription factors binding in this region, the exact same ones. So we thought, how on earth can these be so differentially regulated in these two different cell populations? So we were really intrigued. Oh, that and it's highly expressed in these different kinds of lymphomas compared to normal B cells. So this is just a blown-up view showing that there's this divergent expression. So you've got RNA-seq of the B cell Ig receptor that's highly expressed in B cells, and then in colon there's expression of the IgA receptor and no expression. So in tonsil and colon, which are normal. Now, if you look in the lymphomas, you've got some expression of this IgA receptor that should only be expressed in the epithelial cells. And we thought, what is going on here? Is it because these enhancers are abnormally activated in the B cell lymphomas? So I got a little ahead of myself and already told you some of this, and so I'll just move on ahead. So this is more expression data demonstrating that expression of FCMR is increased in the lymphoma cells, and particularly in CLL, and much less so in normal germinal center B cells, although it is somewhat increased in activated tonsil B cells, which activated B cells and lymphoma cells have many similarities, as you probably know. But then the IgA receptor has this, again, very much upregulated compared to normal B cells, which basically this is, a log 2 of 3 is basically no expression. And then importantly, we had multiple cell models of lymphoma, and then this is an adenocarcinoma colon cell line that also is showing this divergent expression pattern that we could use as model systems for experiments. So, so Jared, my student, started to define what he thought were enhancers in this region, and as you can see, there are many that are intronic, and so there's the DNase is I don't have any histone modifications in this particular slide, sorry, but the DNase also marks the enhancer regions. Just not the full extent of them. But then in terms of locus architecture and how things are looping, right? So there's so looping of enhancers to promoters, or looping out of enhancers or promoters to control gene expression. So this very physical 3D kind of element of gene regulation. And CTCF is a big part of that. And so based on where these CTCF sites were, and some differential binding of CTCF in colon compared to B cells, and then also DNase, and then POL2, which is of course binding for transcription. we came up with this model of looping, of where you could have an interaction of these two regions and loop out, one or the other promoter in the different types of cells, and then isolate it from the enhancer, because the enhancer region, a great deal of the enhancer region is shared, and so looping it away from the promoter and isolating it could be a means for regulating expression. So we've also done luciferase to try to confirm some of these enhancers, and I apologize, there's a little bit of weirdness in how he numbered them, but their location in the locus is numbered here, and the ones that are seen as active in B cells are blue, and epithelial cells are yellow. And so you can see that this enhancer region number 3, which is right here, unfortunately, at the beginning, at the end of the PIGR gene, but overlapping it, is unfortunate in terms of being able to CRISPR it out, is a very strong enhancer, and actually more so in the colon cells than in the B cells, but still very high. And then the others have more or less cell type specificity. Some of them are higher in the B cells, and some of them are higher in the epithelial cells. And that gives us an indication of which are more important for driving expression in these different lineages, and then also tells us which ones we can either cut out and what we would expect to see in terms of expression changes in these different lineages, and which transcription factors really are important, because I said the literature is completely overlapping in terms of the transcription factors that are driving expression of these two genes. But part of that is because they sort of have looked globally and have not actually dissected each of these enhancer regions. So these are the studies that we're working on now in terms of knockdown and overexpression, and looking at different patterns of immunofluorescence and localization, mostly in terms of signaling of FCMR, because that has not been worked out very well, and its potential effects on survival and proliferation in lymphoma. So those studies are still ongoing. So the next story that I want to talk about, switch gears a little bit and start talking about T-cell lymphoma. So we were really fortunate to have some great collaborators in medical oncology who had an ongoing trial for patients with cutaneous T-cell lymphoma, which includes mycosis fungoides and also Sézary syndrome, who were getting romidepsin or vorinostat, because this disease entity is one of the very few that has a histone deacetylase or any histone-related epigenetic drug that is FDA approved for its treatment. And so because of the great collaboration that we have, we were able to get patients pre-treatment, and then at various points in time after they had been treated. And it's FDA approved, so of course if it's FDA approved, you must think, oh, it must work for these patients. And it does in a subset of them, which is of course not great for them, but it sets up sort of a natural experiment for us in that we can compare then the patients who respond versus the ones who don't, because we have those serial samples and that linear progression over their disease course. So we have multiple time points of blood and some skin biopsies. Pre and post treatment, of course, we isolate T cells, and we developed a ChIP-seq method where we could use a very, very small number of cells, because as you can imagine, we don't get that many T cells from a skin punch biopsy. But we were able to get that to work very robustly with 50 to 100,000 cells, which for ChIP is a pretty small number. So when we looked at these patients again, we saw this really striking separation of the patients who they responded to the drug, they were sensitive to the drug, compared to those who were resistant or progressed. And the coloring in this is a little bit different from maybe what you're used to seeing. Green is high activity in enhancer regions, and so the patients who were resistant or who progressed had a huge number of enhancer regions that were active, much more active as compared to the patients who responded. And again, those correlated or corroborated nicely with neighboring gene expression as well. And so I was just going to show you one example. This study is ongoing. That one of these was BCL2, which makes sense. It's very well known, particularly in B-cell lymphoma, right? It's a translocation partner, but it is just more widely being recognized as an important driver of cancer and proliferation in other malignancies as well, probably at least in part because there is an inhibitor, venetoclax, which has been really pretty successful in B-cell malignancies, and I think was actually just FDA approved for certain subsets of AML, which you don't usually think of AML as being driven by BCL2, but it's a anti-apoptotic protein, and so I guess inhibiting it, does work. I think it's patients who have relapsed. It's obviously not a first line for AML, but basically we see that the patients who are resistant have, and BCL2 is a huge gene, so that's why this looks so large. But they're intronic enhancers that are have much higher levels of acetylation as compared to, and this is K27AC or H3K9 K14 AC, which is a little bit less specific of whether it's enhancer or gene body, but it is always activating. And so we see high levels in the patients who are resistant. And BCL2 itself is very highly expressed in the patients who didn't respond to either romidepsin or vorinostat. So our next steps are to continue to expand these studies. And we're adding in some clonal evolution studies because we want to know at the level of the genome, are there differences between these patients? And because obviously we want to understand the mechanism, but also because we want to know, are there any targetable mutations? And also, ChIP-seq is not the best assay for clinical monitoring, as you may expect. So obviously, a genomic mutation would be much easier to use for determining which patients might respond or not respond, or following them over time. So that's what we're doing with the sequencing studies. And then one of my collaborators at WashU is developing PDX models of CTCL, and we're very excited about the potential there in terms of models for potentially therapeutic development or testing, even of like existing therapeutics, so like venetoclax. And then, of course, our cell models for looking at more mechanistic biology. So then the last project that I want to tell you about is link RNAs. And link RNAs aren't just for enhancers anymore. So we initially became interested in, and I think I actually took this slide out, but we initially became interested in long non-coding RNAs because when we were trying to define enhancer profiles, and I showed you in one of the first slides, enhancer profiles that were very different between follicular lymphoma, that was the more responsive type, the germinal center type of follicular lymphoma, and the type that had more of an activated B-cell profile, and those patients did worse. Those enhancer profiles were very, very different. And so we thought, well, obviously, follicular lymphoma, which is very indolent, will also have a very different enhancer profile from diffuse large B-cell lymphoma, which has a very different clinical progression. And it proved to be actually fairly difficult to find enhancer profiles that differentiated those two entities. And I think that a big part of the reason is that DLBCL tends to be a very heterogeneous tumor. Of course, there's GCB and ABC subtypes, but then there are a number of gradations morphologically and expression-wise in addition to that. And so we were really struggling with it. And then finally, my postdoc found, she found she found a set of enhancers that actually did differentiate them. And we thought, wow, that's great. How did you find those? What is different about those? And what was different was that they were near or overlapping with highly expressed long non-coding RNAs. And this was, a few years ago, and we were like, what is even a long non-coding RNA? What is this thing? And so we started investigating them. And so a very similar initial kind of plan of study, but then we wanted to differentiate between long non-coding RNAs that were known and annotated and those that are novel, because probably is not that surprising to you that long non-coding RNAs are similar to enhancers or to superenhancers in that their expression patterns are very lineage specific. So if you look at mRNAs, there's a small percentage of them that are only expressed in certain cell types, but it's a very small percentage. Whereas if you look at long non-coding RNAs, a very large number of them are only expressed in a certain cell type. And that probably is largely driven by the fact that many of them are expressed from enhancers. The annotation and definitions of long non-coding RNAs is a fairly, people argue a lot about how to define all of these. And so again, I'm sort of agnostic about it. I try to be more descriptive, so I hope I haven't offended anyone in the room about how I'm describing links. But that was our goal in trying to identify novel transcripts that were only expressed in B cells, and then also the annotated transcripts, and also with an eye to the fact that there may be links that are only expressed in lymphoma, because those obviously would have escaped all of the encode, annotation, and all of that. So we compared the links that were expressed in lymphoma to the ones that were expressed in normal B cells, in a kind of a typical volcano plot. And there were, almost 1,000 that, met our cutoffs for being differentially expressed. And as I alluded to, my lab is too small to functionally characterize 1,000 link RNAs, especially when you consider the fact that there are a huge number of functions that have been ascribed to different links. And everything from different mechanisms of gene regulation, many different mechanisms of gene regulation. So recruiting chromatin modifiers, sequestering transcription factors, actually looping of enhancers to promoters. I mean, many different, just in the nucleus. And then, at the time it was more recent, cytoplasmic mechanisms and functions that link RNAs have. And, these were a huge number, so, involved in stabilizing mRNAs or as a sink for microRNAs. There's quite a bit of literature of link RNAs as sinks for microRNAs. Circular RNAs, which is a whole different field, and acting as scaffolds for different proteins, bringing them together so that they can phosphorylate each other and then participate in signaling pathways. So it was overwhelming. And so our solution was that we wanted to develop an algorithm, a bioinformatic approach to help us to prioritize and zero in on potential molecular functions of the links that we found that were lymphoma associated, so that we could better decide which ones should we pursue and how should we pursue them, that being like the biggest, most important question. So this is Plato. Plato was the tool that we developed to solve this problem. And my student Sarah designed Plato, and this was her favorite part of it, which was the heuristics part, because she really wanted to emphasize the fact that while there are algorithms and calculations of scores that predict the function and the relative likelihood of a function for a link RNA, at the end of the day, it still requires a person evaluating this data and making a decision, because the goal is to find something that you're going to then do experiments on, and that requires a person. So we start with this idea. So Plato starts with link RNAs as potential gene regulators. And probably the majority of them are involved in gene regulation if they have a function. Maybe not all of them do, or we don't know yet. But so, and presuming that they may have local gene regulatory function. There are link RNAs that act on genes, regulate genes that are distal to them. But given the data that we had, which were relatively small clinical samples, and we also, observed, and from talking with our colleagues, most people's studies are relatively small experiments, whether they're done from human samples or whether they're done from, mouse experiments in their lab. They're relatively small. And so to have the power to reliably and robustly and statistically significantly detect regulation of a distal gene is a pretty tall order. And so we focused on local regulation. And we incorporated the same kinds of data that I've already alluded to, and also considered RNA binding proteins and importantly, subcellular localization of the link RNA. And this is just super quick overview that I will go into more. Anyway, these are some of the outputs of Plato, and I'll walk you through these. So the first thing that Plato assesses is what is the likelihood that this link RNA is involved in the regulation, the expression of this next gene, and it can go out. It's a very flexible tool, so you could put in any distance that you want. In this particular study, we went out 400 kb on each side because that's the size of an average TAD, topologically associated domain. So from all of those assays that I showed you a minute ago, Plato calculates a score, okay, which involves K4ME3, the correlation of expression between the link RNA and its potential gene partner, whether it is nuclear or cytosolic in its fraction, and calculates what we call the link RNA transcript cisregulatory score, which just means what is the likelihood that this links, the transcript itself, is important for the expression level of its linked gene. And it does this in a pairwise fashion. So you can look at any link RNA coding gene pair. And so just as an example, so we did these in ENCODE cell lines, which have a lot of data and have been very well studied. And so they were a good initial kind of training set to test Plato. And we found that there were a number, and I'm just showing you some examples, of hot air and ZIST that had a score above our cutoff, indicating that those link RNAs are more likely to regulate the linked, or the nearby gene. So the next thing that we wanted to figure out is the kind of flip side of that, which is, no, this link RNA just happens to be expressed from this enhancer, and it's actually the enhancer that's important for regulation of expression of that coding gene. It's really not the link RNA transcript. And this is a constant argument, but real biological question and problem in the field. And so we wanted Plato to give us an indication of the likelihood that no, actually it's just the enhancer. And so the way we did that was we calculated a different score, which was the H3K4ME1, which I told you at the beginning of the talk is a mark of enhancers, whether they're active or not. So is it even an enhancer? Is this link RNA being expressed from an enhancer? And then how active is it? How much K27 acetylation does it have? And then we were using ChIA-PET, which is a measure of chromatin looping, to say, is this region that the link RNA is in, is it actually looping to the gene that we are trying to determine whether it regulates the expression of, okay? So that went into the score calculation for the enhancer cisregulatory score, and we found a number of links that are expressed from enhancers that are known to regulate the coding gene. So this was actually confirmed in a link RNA screen that was published last year. This one was confirmed in another link RNA screen from the same group. And then this one is a little bit, has been known for longer, but is confirmed in the literature. So we combined these two and plotted them against each other so that we could isolate which lincRNAs have a purely high transcript cis-regulatory score and which have a purely high cis-regulatory score, because the ones that score highly for both, we don't necessarily know what to do with those, and we certainly aren't going to follow up on them functionally because we just don't know which they are more likely to be. And so this approach separated exceptionally well based on validation, literature validations, okay, these different lincRNA coding gene pairs. And so those, I told you those were encoded cell lines. Those are cell lines. How much of that is really real? So we did the same, used the same approach in DLBCL, primary DLBCL samples. Now, not all of these have been confirmed in the literature because this data is not as highly available or well studied because it's not an encoded cell line. But some of them, PBT1-MYC, for example, has been confirmed in the literature, and a number of others sort of make sense from a mechanistic point of view. So we were really, really excited about that, about being able to differentiate which link is actually, the transcript is actually regulating expression. So the next thing that we wanted to do is say, okay, well, but links aren't just regulating genes. They also have other roles, and the easiest way to differentiate that is then looking at which ones are in the cytoplasm. And so we used some publicly available data on subcellular localization of all RNAs. That there's more and more. The databases are growing, but initially it was a little bit of a challenge. And then also looking at a new publicly available source, which is RNA binding protein pull down sort of on a massive scale. And so there's a database of, gosh, I can't remember if it's 200 or 300 RNA binding proteins where they have done pull downs and then done sequencing, RNA sequencing to determine all of the RNAs that are bound to these RNA binding proteins. Now, obviously, it's biased because they chose which RNA binding proteins that they put into their experiments. And they also had to have good antibodies to begin with. They actually did an amazing job of making sure that each antibody was actually specific. But nevertheless, it is a compendium of information that we were able to use. And so we incorporated all of this data and used the additional knowledge of subcellular localization this is the initial step that Plato goes through. So it separates whether the lincRNA and the RNA binding protein, where they're localized, cytoplasmic or nuclear, or cytoplasmic and nuclear, because unfortunately most of them are both. And then, so that can give you an indication of what is the likelihood of these actually being functional, right? Because just because you find it in a pull down, you've popped open the cell, right? So they could bind each other and it could be artifactual. But if they're actually separated in the cell and they bind each other through this experiment, then there's a greater likelihood that it's actually a real functional effect mechanism. So, but then we went a step further and we said, well, we really need to rank these, because if we're going to choose something that we're going to go after functionally, we need to rank them. And so there are a number of ways that we tried ranking them, and this is eventually what we came up with, which is the co-localization is indicated by color. Okay, so if they're both in the cytoplasm, they're red. If they're both in the nucleus, they're blue. And it's really hard to tell, but if they're both, it's purple. So over here, those colors remain the same. The lincRNA expression in log 10 is on this axis, so higher is more expressed. And the RBP sites per kilobase. So this is a measure of how many sites of binding of the RNA binding protein actually measured, not potential or motif or what have you, but actually measured binding sites. There are normalized to the size of the lincRNA, because the definition of a lincRNA is anything that is non-coding that is greater than 200 base pairs. So they can be 201, they could be 10,000, it's a huge range. So we normalize for the size of the lincRNA. And then in addition to that, the size of the dot is an indicator of how highly the RNA binding protein is expressed. So again, if you're wanting to go after and do experiments on something, you want a lincRNA that's highly expressed, you want an RNA binding protein that's highly expressed, because otherwise your experiments are going to be very challenging. So we compared it with the literature and found a number of lincRNA, RNA binding protein interactions that are confirmed and are important for many different aspects of biology. They sort of fill the range. So, you know, TERK and HD19 and NEAT1. We've done some work in confirming Plato's predictions. And so we have looked at a number of lincRNAs that we've identified as being very highly expressed in lymphoma. JPX is a control, so it is known to be a long non-coding RNA that's associated with chromatin. And then, of course, 18S RNA is predominantly in the cytoplasm. And then we identified this is a lincRNA that is upstream of Polino-1. And it is, as we predicted, enriched in the nuclear fraction and is most likely associated with the enhancer that is regulating Polino-1, which is an important immune response protein. And then there are and another one that's also was predicted to be in the chromatin and is, and then a couple of lincRNAs that were much more enriched in the cytoplasm as compared to in the nucleus. And so one of these we were really intrigued by, and we call it lincRNA PLCG2 because in reality it has some mouthful of an RP11 blah, 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 as they tend to annotate these things. And it's, again, you'll kind of see a theme. It's really in a fascinating locus because there's this CMIP gene, which is expressed in T cells. And then PLCG2, which is phospholipase C-gamma2, which is mostly expressed in B cells, very highly expressed in B cells, and is activated downstream of the B cell receptor, which is, of course, extremely important for B cell development and activation and lymphoma. And we saw that this lincRNA was much more highly expressed in the lymphoma cells, although still, somewhat expressed in the normal B cells. And, really fascinating that this CMIP is expressed in other lineages. I don't have a T cell on here, but trust me, it's expressed in T cells. But then PLCG2 is not, and the link is not. And again, it's very much enriched in the cytoplasm as compared to other subfractions of the nucleus. We did a lot of experiments that showed that this link does not regulate the expression at the RNA level or the protein level of PLCG2, its neighboring gene. Knocked it out, knocked it down, never saw any difference in expression. So then we thought, okay, well, what could we possibly look at? Okay, so I kept telling my postdoc that this was never going to work, and luckily for me, she didn't listen to me because she started looking into how does PLCG2 work, and as I said, it's downstream of BCR activation. Do I have a? Yes, I do. So when you activate BCR, at least when we do this in vitro, we add IgM, and then there's a chain phosphorylation of Lin-Sig, Blink, and BTK, which then activates PLCG2 by phosphorylation, which then cleaves PIP2 to diacylglycerol, IP3, which then activates calcium and calcium signaling, which then, of course, then signals to changes in expression through NFAT and other transcription factors in the nucleus. But most importantly, this was a phenotype that we could actually measure, okay, calcium signaling. And so we used CRISPR to knock out the link. And when we measured calcium signaling, and we've got the cells, they're just sort of, hanging out here. We add IgM, and the wild type starts pumping out calcium. This is a FURA2, which just measures calcium flux. Whereas the knockouts are somewhat suppressed in their calcium signaling. We've done this a couple of different ways. One where we tried to isolate the different steps of calcium signaling, so initial calcium flux out, and then calcium flux. If you add calcium exogenously to the media that the cells are in, it will flux back in through the crack and the SOCE, there we see some differential effects on those two processes, but we're still trying to sort that out. So as far as ongoing studies, obviously we're very interested in looking at what is interacting with this lung non-coding RNA, and so we're using different approaches for that, as well as looking at structure function. So just, super quick summary, enhancers are really important and they drive cancer pathogenesis and progression, and we are trying to define not just novel mechanisms but novel markers that we can target for therapeutic development and to better understand how gene regulation works and becomes perturbed in cancer. I just wanted to thank the people who actually did the work. This is my lab. Last summer, we went to the City Museum in St. Louis. If you ever visit St. Louis, the City Museum is awesome. It's so much fun. It has like a school bus and a plane on its roof, and you can climb through them. Very cool. Jared is the one who did the lion's share of the first two projects. Sarah is doing the bravely continuing the link RNA work that was started by my postdoc Hong. And then our longtime collaborators in the Oltz lab, HemePath, of course, our clinical collaborators in oncology and dermatology, and of course our funding sources. And most of all, thank you for your attention and your time.
About this second project where the levels of enhancer in the lymphoma were always high, or it was altered after HDEC-I treatment, so when in the resistant cell it was always high, or
right.
There was alteration after?
Right, so I wasn't able to show the data, but we actually did compare those profiles to cells that we had treated in vitro. So both, we have CTCL cell lines, and then we had normal T cells that we actually purified from ourselves. And we compared, so an HDAC inhibitor prevents the removal of acetylation, right? And so the profiles that we saw in the patients who were resistant were not only the opposite of the patients who responded, they were the opposite of the samples that we treated in vitro. And so the difficult thing about HDAC inhibitors, at least the current generation, the ones that are being actually used to treat patients, is that they are not very specific. They will inhibit the deacetylation, any of the deacetylases. The problem with that, so you don't know which histone modification they're going to be affecting, but you also don't know if they're actually affecting other proteins. So it's not just proteins, histone proteins that are acetylated, it is many other proteins. And so if the most important protein that they are affecting is actually not a histone protein, it maybe is a protein whose downstream effect is acetylation of histones, then that's sort of an indirect route that we're still trying to work through. So the simple answer is all of the patients who progressed had the opposite profile compared to the normal samples or the patients who were sensitive. Mm-hmm.
So in the first part, you were talking about the super enhancer. Oops, sorry. In the first part, you were talking about changes specifically at the enhancer, right? Mm-hmm. Did you see changes everywhere in the genome, or specifically just at this enhancer? And what's your idea of what causes that?
Oh, in comparing so the first project, the follicular lymphomas, the two different types. So we were particularly interested in the enhancers, but we certainly saw changes also at the promoter. Not always at the same gene, often at the same gene where the enhancer profile changed. We were not as interested in looking at the promoters because that seemed sort of obvious to us, and so we focused more on the enhancers. If you mean regions that are not enhancers, or like, you know, did
you see any overall change in the pattern of over the genome? Or it's very difficult actually to quantitate.
That's true. That's true because then you don't know
because
if it's just people just assume that it's kind of noise.
Normalized, yeah.
Right. People just assume that it's noise elsewhere, right? Right. So we did do, again, I didn't have time to show the data, but we did use other histone marks to define what was an enhancer, or open chromatin marks to define what was an enhancer, and required that those also be there to define that as a way to sort of approach this question. In general, though, what we excluded by making those requirements was, for the most part, was genic regions. There were very few regions where we saw patterns of histone acetylation that didn't also have, open chromatin by DNA surfer attack, or H3K4ME1 or what have you. They pretty much coincided almost perfectly. So.
Thank you.
Mm-hmm.